Hello, everyone, and welcome to this Cytognos webinar co-hosted by Biospecifix, our partner in Australia, New Zealand, and Hong Kong. My name is Guadalupe, and I work as product specialist at Cytognos, and I am presenting today your cheat sheets for smarter clinical flow cytometry testing. I have Steve from Biospecifix today. Hello, how are you?
Hello, Guada. Lovely to hear your voice. I'm looking forward to the presentation today. Thank you very much for this webinar. I think it's going to be very succinct and explain a lot of things very well to our clients, especially to newcomers to Euroflow and next generation flow cytometry. So thank you very much, and fire away.
Thank you, Steve, for your introduction. So now we are ready to start. These are the points we are going to cover today. First, I will start off highlighting the value of flow cytometry in clinical practice, and then I will talk about the current issues we face in the clinical routine lab, and I will focus on the specific challenges of the minimal residual disease assays and on the design and validation of flow cytometry tests. And finally, we will see how we can overcome these challenges with a fully standardized solution that is the Euroflow method. And at the end of the webinar, we will have a few minutes for questions, so you can write them on the question box you can find on the right part. So what is the role of flow cytometry immunophenotyping in the clinical lab? It allows the diagnosis and classification of leukemias, lymphomas, and plasma cell dyscrasias, as well as the detection of minimal residual disease after treatment. But also flow cytometry is used for studying the immune response of patients and for the progress control of the treatment itself, and in the clinical immunology field to support primary immunodeficiency diagnosis. So flow cytometry is definitely a versatile and powerful technique with many applications in clinical settings, but it's also complex and it has multiple steps. So first, we collect the specimen and transport it to the lab. Once it gets there, we process the sample and we stain it. And acquire the tubes in the instrument, which has been previously calibrated and compensated. And in the last step is analyzing the data, and we report the result to the clinician. So there are multiple checkpoints here where different errors might be currently being introduced and ignored, and especially in the pre-analytical and post-analytical phase. And these errors have economical consequences and negative effects in the patient care, right? So that's why all flow cytometry assays require careful assay design, instrument setup, performance validation, and ongoing quality monitoring. Now let's see what are the common challenges we face in clinical routine. Regarding sample collection and transport, first, sample stability can be an issue. Most of the times we require a fresh sample, in some cases no longer than 36 hours after collection. And the samples that come in are very common, very diverse, so we get blood, bone marrow, lymph nodes, and they come with different anticoagulants like EDTA or heparin, for example. So they require different sipping and different storage conditions, especially with regards to temperature. For sample processing, there are different workflows protocol in place, so we have to coordinate multiple workflows at the same time. Considering sample staining, cocktailing the single antibodies can lead to pipetting errors. And for example, if we get a new lot, we need to cross-check it with the previous one. And we need to have an efficient stock control of all the reagents if we want to reduce the reagent waste. All of our instruments have to be correctly set up, and we need to track the day-to-day performance to ensure consistent data over time. I mean, reliable and reproducible data over time to ensure that the instrument is performing consistently. For example, the optical alignment, fluidics, laser power, and the PMT voltage must also be monitored. It's important also to acquire the tube as soon as possible to minimize sedimentation and not to compromise the quality of the sample. And during acquisition, we can run into issues such as unstable flow, clogging, or even air bubbles. When it comes to the analysis, there is always a user bias because positive and negative population are clear, but there is a lot of middle ground in between. So the final result highly depends on the analysis expertise. Lastly, we have to put together a report with our lab test results, transfer it to the LI system so it can be accessible to the pathologist or clinician, while ensuring that all the sensitive data is protected. So those are common points regardless of the type of study, but however, more sophisticated tests such as the minimal residual disease pose additional challenges in the lab. So now let's take a look over that. For example, for the minimal residual disease assays regarding sample collection and transport, to follow a patient, we usually analyze bone marrow, which is a delicate specimen, as we know. Sometimes we only get a small volume to run the test, and we can have hemodilution issues if we don't get the first pull. Moreover, the anticoagulant also may affect marker expression. For example, heparin downregulate the 138 expression in plasma cells. Then we have to bulkize the sample up front to concentrate the number of cells, so we need to add an extra step to our sample processing protocol. We need as well to make sure that our MRD panel are optimized for detecting the target population at very low levels, and our MRD panel are adapted to immunotherapy that can interfere with the expression of a specific marker. And definitely, we need to make sure that our instrument has the capacity to collect a high number of events relatively fast. Here, one of the biggest challenges with our MRD assays, there are the false MRD negatives. So we are handling with a large number of events, and we want to identify a population that has a very low frequency. So high sensitivity is key to avoid reporting false MRD negatives. So plus, we need to consider the patient treatment for the gating strategy, as some immunotherapies, as just mentioned, can mask the expression of a specific marker. On top of that, in every pathology lab, we are getting more and more cases. We have a very tight turnaround time. In addition, because of the COVID-19 pandemic, hospital may have cut the budget for the lab. And at the same time, we need to meet all regulatory requirements for diagnostic we have for the diagnostic lab. So let's summarize all this in one sentence. In a clinical lab, we have to get results out quickly without compromising the quality. Now, before introducing a new test in the lab, we need to carefully design and clinically validate the assay. So to design a new test, there are several aspects we need to keep in mind. First of all, it's the clinical question we want to answer. And this is going to determine the panel design, the sample processing protocols, the instrument settings, the data acquisition parameters, and for sure the data analysis strategy. And once we come up with the antibody panel, we need to test the entire assay. And this is an iterative process that can take many rounds of design, redesign before we complete. After we are done with this testing phase, we need to ensure that our assay complies with the current in vitro diagnostic regulation. So we need to put together a fit-for-purpose validation plan that confirms that the performance characteristics of our assay are acceptable for the intended use. And for that, we need to assess several parameters like specificity, precision, sensitivity, which is essential, for example, in the case of MRD assays, stability, carryover, and even establishing reference range. Today, there is no specific guideline for validating flow cytometry tests in regulated environment, and it makes the entire process more complicated. So the question is there a way we can get consistent results over time across instruments and operators, save money and time while we comply with the regulatory bodies? The answer is yes, by standardizing all steps in our clinical routine. Okay, that's exactly what the Euroflow method is about. The Euroflow Group is an independent scientific foundation made up of over 20 companies and research groups, which was born in 26 with the mission of developing, standardizing, and validating highly sensitive flow cytometry tests for diagnosis and follow-up. Okay. So the Euroflow method is a screening approach looking to use figure antibodies at first to narrow down the malignancy. Those are the screening tubes, and then a running classification panel for further differentiation. They started with the leukemia and lymphoma panel on the left part, followed by the MRD panels to monitor patients. And once they got this system in place for onco-hematology, they apply all their expertise and knowledge for the primary immunodeficiency and for the immune monitoring field. So this method can be used in the onco-hematology for clinical trials also, and in the immunology area. This method includes the standard operational procedure, the antibody panels, the analysis software tools, and the QA program. So let's go over each of them. There is a protocol for bone marrow sample collection and transport, specifically for myeloma MRD, but also there are guidelines on sample collection for regular testing. Euroflow also provides a protocol for sample processing and staining, including a detailed explanation about extracellular and cytoplasmic antibodies staining, and other more specific, for example, regarding the TDT marker. There is also a specific bulk license protocol for MRD testing. For building leukemia, lymphoma, myeloma panels, Euroflow tested a large number of fluorochromes and antibody clones. So they selected the fluorochrome based on the MFI, the stain index, the spillover, the stability, and the availability in the marker. Here we have an example illustrating the comparison between two fluorochromes, the APC-H7 and the APC-C750, conjugated with the same antibody clone, the CD10, and tested in the same sample under the same condition. We see that the APC-C750 shows a brighter MFI, which provides a better resolution between the positive and the negative population. Likewise, for selecting the marker, they look at the antibody clones, what is the best conjugate, also the number of combined markers in a single tube, and the amount of antibody and the volume of stain. They tested the performance of the marker combination together using software tools, and that's what we can see in this diagram. So these are multidimensional plots showing cases of TALL in green, BCP-ALL in blue, and AML in red, staining with the Euroflow acute leukemia screening panel. In each plot, they left out one of the markers in order to study one by one the discrimination power in these three acute leukemia subtypes. So these results indicate that each individual marker, including the panel, is essential because missing one of them affects the identification of the different acute leukemia entities. There are protocols for instrument setup, calibration, and compensation available for different instruments. This protocol covers the instrument configuration, the setup of forward assay scatter, the setup of the PMT voltage for the target fluorescent channel using the rainbow bead, the compensation settings, and the instrument performance monitoring. For our data analysis, we need a flexible and advanced tool that make the multicolor analysis easier. So, for example, by turning off the background to just focus on the target population, by looking at our data in many different ways, also we can study the cell maturation path of a specific population. Also, we need tools to simplify the multi-parameter analysis. As flow cytometry technology evolves, there are more and more parameters to analyze. For example, here we have all the possible combination of marker in an 8-color panel that is 64 diagrams. So instead of looking at all these 2D plots, we can convert all of them into one multidimensional diagram that separates out all population using all the parameters we have in our panel. And this way we can discriminate population with a very similar phenotype that can go unnoticed if we just look at the 2D plots. We can check the complete immunophenotype of a target population versus the normal control. But also we can follow up patient with confidence by loading the diagnostic population onto the MRD case as a reference. Using advanced tools, we can analyze patient case, not single tube. And this is very important as we can combine all the tubes from the panel, analyze them together and create one single report. So we are getting one case, one report. But still the problem in flow cytometry analysis is the lack of a good reference point or baseline. And as complexity of flow panel and data analysis increase, we really need big data analysis approaches and we need automation for our clinical flow analysis. To put it simple, the automation consists of clustering all the events in the file, followed by an identification of all these clusters. But to be able to give a clinical and biological meaning, not only to characterize this population, but also for the interpretation of the results, we need to feed this algorithm with human knowledge and expertise, and that is the Euroflow reference database. Okay, so let's see an example of automation in acute leukemia screening. We get here all the cell subsets from our sample identify and characterize. We get a compass diagram showing the cell lineage, I mean the BET or myeloma that the aberrant cells are more likely to belong to. This is done through a multiparametric comparison of your sample versus the reference database containing B in green, T in blue, and myeloid leukemia cell in red. Here we have the interpretation of the case, including not only about the blast, but also the detection of potential related disorder. You are going to have the complete immunophenotype of each of the aberrant population, even if you have several lineage affected. Additional information or the present or missing population of the patient. And most importantly, which panel to run next for further classification of the disease. Now let's see an example of automation in myeloma MRD. Here we can see the identification and characterization of both normal plasma cell and abnormal plasma cell after automation. Here we have the interpretation of the case, including the complete immune profile of the patient versus the normal reference, information of bone marrow regeneration, the MRD level and the immunophenotype of the aberrant population, the clinical threshold that determines the sensitivity of the test, and information on sample quality, issues like hemodilution or sample viability. Okay, so let me back to the step of flow cytometry assay. And last but not least, UFO runs 2 external quality assessment programs to certify that the consistency and the reliability of results generated in the lab. So we have 2 QA programs, the LSTQA and the MRDQA. The LSTQA monitors the overall flow testing performance, including the sample preparation, the instrument setting, and the data acquisition. And in the case of the MRDQA, this program specifically targets the data analysis of the myeloma MRD assay. So the Euroflow goal is to provide standardized and validated, highly sensitive flow cytometry tests for diagnosis and follow-up of patients, and that requires a close collaboration between industry and academia. And thanks to this collaboration, Cytognos can beat the Euroflow method as a ready-to-use flow solution for any lab in the world. This is what we call next-generation flow solution. And this solution includes the laboratory SOPs, the dry-down antibody kits, the instrument, and the Infinity software, and ongoing training and support to help you along the way. So to wrap up, these are the key takeaways from the webinar. Flow cytometry is a powerful and versatile tool in clinical practice and yet complex. Standardization is the way to obtain reliable and reproducible results, and the Euroflow method is available through Cytognos as a ready-to-use solution for your lab. You will have our contact information here. Thank you for listening. Thank you for watching. We can now open the question session. Thank you.